E ngā mana, e ngā reo, rau rangatira mā. Tēnā koutou, tēnā koutou, tēnā koutou katoa. Nau mai, haere mai, ki te whare pūtai ao. Ngā mihi mahana, kia koe Maren. It's an absolute treat to be here today to introduce you for this really auspicious occasion. Ngā mihi kia koutou, ngā mihi ki tō hoa mahi, plant and food research, me te whare pūtai ao. It's lovely to have people here today from plant and food research in Auckland, but also we've got people from Whakatū, from Nelson. Where are our Nelson people today? Thank you so much for coming. It's just a marvellous occasion to be able to celebrate Maren's success in this way, and it's great to have you up here. Ko JR hau ko te manukura rua o te whare pūtai ao. My name is JR and I'm the Deputy Dean in the Faculty of Science, and I can say that today because it's the first day of our new Dean. Last month I was the Acting Dean, and it's lovely to go back to being the Deputy Dean. One of the greatest things, the greatest pleasures of being a Deputy Dean or a Dean is that you get to introduce inaugural professorial lectures. And they are such a treat because getting to be a professor is the absolute hallmark of an academic career, and it is an incredible testament to what you have achieved, Maren, that the Faculty of Science was able to support your application for promotion, and the University of Auckland recognised your achievements, and here you are as a professor. Tēnā koutou, tēnā koutou, tēnā koutou katoa. It's just so lovely to have everybody here making this room warm. Now, it's my custom in introducing inaugural lectures just to shine a light on a few of the things that I think make our professors outstanding. And Maren, I just want to hone in on the fact that you are one of our joint graduate school professors now on a part-time position at the University of Auckland, generally based in Nelson in plant and food research. And it's incredibly difficult from these roles to actually get promoted to be professor. So I think there's an extra moment of celebration in here to congratulate you on that, because it is a very, very challenging thing, because you don't get to have all the time and largesse that the rest of us have in the university sector to be able to meet all of the academic standards. And the fact that you've done that is absolutely fabulous. So I just want to hone in on a few things, and I'm going to start with the teaching, because as a joint graduate school academic, one of the key aspects in that role is the supervision of students, and to bring students through the university and to share what the Crown Research Institute can offer and all that you bring to that environment. And in your time with us, you've graduated now 11 PhD students, possibly more since your application went in, 6 master's students, 7 honours to completion, and you've also supervised another 9 postdocs, which is an incredible contribution to our research community and to society. You've had large numbers of summer students and interns. In particular, you've had a particular focus on Māori and Pacific Island students as well. You've been mentoring people through into the science sector, and as I said, that is an enormous contribution to society. You don't have to do taught classes necessarily as a Crown Research person coming into a joint graduate school, and you have over-delivered. Alan, I didn't really mean that, because we really love what you're doing in that delivery, but you have delivered exceptionally in terms of your contributions to the taught curriculum in the School of Biological Sciences, and we are very grateful for the impact that you have had in terms of scaffolding the evolution education through particularly that undergraduate curriculum. Aside from the teaching side of things, Maren, you're an absolutely outstanding researcher. You had, at the time of your promotion application, 106 publications with outputs in world-class venues, including Nature Communications in your field, over 3,500 citations. You are a lauded academic researcher with a Thermo Fisher Scientific Award for Excellence in 2020, the Royal Society Te Āpārangi Hamilton Award 2018, Science New Zealand Early Career Researcher Award in 2017, and the award that I was the most intrigued by, the KVA King Carl the 16th Gustav's Personal Award from the King in 2013, which sounds absolutely exciting. Not only that, in terms of metrics, your granting success is unbelievable. You've had over close to $25 million secured in funding, over 38 grants, PI on 33 of these, prestigious grants from Marsden to MBIE, Swedish Research Council, European Commission. You're a star in the research space, and we are very fortunate that you have formed a very strong relationship with the University of Auckland. You're also a wonderful mentor, and we really appreciate all of that mentorship work you do with our students and our staff. And as befitting of a professor, you are a marvellous citizen and leader in this university. You have an international reputation via your editorships, your network of organisations that you belong to, national responsibilities and co-chairing, for example, the PBRF panel for biological sciences. You're a group leader in your own institution and have been for some time at Plant and Food, and you locally contribute into things like the School of Biological Sciences Marsden Club. An all-round amazing citizen, fabulous researcher and wonderful contributor to academia, and you thoroughly deserve your promotion to Professor. Please join me in congratulating Maren on her very, very worthy promotion. And I deliberately haven't talked about your story because you're going to do that. The floor is yours, Professor.
Thank you so much. Thank you so much. Kia ora koutou. It's really nice to see so many friendly faces here, and I truly appreciate you all coming. So for those of you that have not been to an inaugural lecture before, these are, as you heard, for newly appointed professors to share the story of how they got to where they are now. So in short or in brief, like I will basically be walking you through my life as a scientist to explain how I got here. And I have to admit that I initially struggled a little bit to find a title for my inaugural lecture, because when you look at my research path, at least from a distance, you may think that I've been working on quite different research themes. But I think that once you dig a little bit deeper, you may realize that they are connected by a common theme, and that is really a desire to understand the building blocks of biodiversity or the threads of biodiversity. And that's why I thought that unraveling the tapestry of life is actually quite a good metaphor for my scientific path. Now, I thought before we get into the different sort of scientific chapters that mark my own scientific journey, I would like to go back all the way to the beginning of where it all started, my childhood, to actually understand what made me want to become a biologist in the first place. All right. So here you see me with my family on a beach holiday in Denmark. So this is me with my mom and my dad and my brother. And on the right, you see me a little bit older, sporting a haircut courtesy to my dad. Now, my parents really raised me to be like a sort of friendly, easygoing and confident child. And I played for most of my childhood sort of in the outdoors, in the garden and in the woods. And both of my parents are really big nature lovers. My mom is an avid gardener, and she's still spending most of her days now tending to her plants. And my dad also made a real point of spending a lot of his free time in the outdoors. He would often like to create these biodiversity hotspots in my mom's garden, much to her dismay, because they really looked like areas where someone had forgotten to use a lawnmower. But he also really liked to go on big walks, and he would often take me with him on these walks. And he would always try to spark my curiosity and get me to ask questions. I never planned to become a professor. It wasn't part of my life's roadmap, and I think that is probably true for the majority of professors. However, looking back at my childhood, I do have to say that I was raised really among teachers and university lecturers and professors. So I think you can actually say that I come from a truly academic household. My mom and my dad. My mom actually, she earned a master's degree in majoring in pedagogy and German, And then when she had children, she became a teacher. But while she was studying towards her master's degree, some of her fellow students said that she should attend the lectures of Maren Wellenreuther, my dad, and the rest is history. Now, my dad, he got a PhD in sociology, and he was, his lifelong journey was to understand how students learn and how to best teach. And like so many academics, he was very passionate about it, and he would often bring home his newest ideas, something he read, the new theories, something like that. And just to give you a little bit of a flavor of the sort of things that he would bring home to us to discuss with us, I show you some of the books that my father published during his career. And because not all of you are fluent in German, I will translate them. So from left to right. So on the left, you see one of his books, Forschungsbasierte Schulpädagogik, which means as much as research-based school pedagogy. Then you have Lehren und Lernen, aber wie, which means as much as teaching and learning, but how. Then we have Bildungstheater, which means as much as educational theater or theater of education. And then my favorite, quantitative Forschungsmethoden in der Erziehungswissenschaft, which means as much as you can, quantitative research methods in educational science. And I think you can see two things there. So one, he truly really was keen to understand, as I said, how students learn and also how to best teach. But I think a more important theme that is emerging, and that's been really relevant to me and my upbringing, was that my father, his whole approach to his work was really empirical and really research-based. And that actually set him quite apart from the majority of his colleagues, particularly at the time. And my dad, I would definitely say, he was quite fiercely empirical and also fiercely skeptical. And this is why I think the next photo of my dad quite nicely shows his spirit. So this was a photo that my dad asked me to take of him. We were walking through Washington, D.C., and we walked past the National Academy of Sciences, and he saw this quote by Albert Einstein, and he read it. I read it out to you. The right to search for the truth implies also a duty. One must not conceal any part of what one has recognized to be the truth. And he connected with that, and he said, Maren, take a photo. Of course, for the rest of our trip, we went to the Smithsonian Museum for Natural History, and there we really bonded over our shared love for nature. And we looked at, butterflies, examined skeletons, and sort of went through the greenhouse. We were captivated by all the plants. And I really think it was this dual nature, this dual connection, the love that my parents had always shown me for nature, but combined with their quite empirical way to view the world and being quite research-focused all the way through. And so I still remember my parents often shaking their head and laughing and saying, so funny, right, that we, two parents with a background in education, gave rise to two children that have become scientists. My brother has a PhD in physics. He works at a synchrotron, like a particle accelerator. So yes, but I think for me it was the only way. For me, biology was the way to go. And so I knew actually from quite a young age that I wanted to become a biologist. And I still remember when I was 15, I said to the rest of the world, I want to become a biologist. And so I went to, I was raised and born in Göttingen and went to school there. And right after I finished school, I went straight to the university in Göttingen. And it's a great university town, 100,000 people, over 30,000 students, really buzzing. And I immersed myself in biology. So I did my bachelor's degree there, and I loved it. And I also made really good use of my semester breaks. On one of them, I joined an expedition that was going to Borneo to learn something about amphibians around the Mount Kinabalu National Park. And that's me joining that expedition, and I spent 2 months there. But really importantly, I also learned during that trip of how to dive. And that was one of these life-changing moments for me, because I dived and I saw kind of the life under the ocean surface. And it was one of those moments that just went, and when I ascended, I knew two things. I knew I was really in love with the ocean, and I also knew I wanted to become a marine biologist. So after that, I sort of sprang into action. And I moved from Göttingen to the University of Hamburg, and I focused on marine biology, majoring in fisheries and hydrobiology. And it was fabulous because we were a very small cohort. We were very tight, and we got to go out on these research expeditions, these big research surveys that were run by the European Union, or these European sort of research vessels, like here, the Walter Herwig 3. And we spent weeks and weeks out on the ocean, being sort of involved in fisheries surveys. And I loved it, even though my tummy didn't quite agree with it. And I also, though, learned during those trips that I had a real interest in better understanding more the sort of the interactions between the different species and also species in the environment, so ecology. And so knowing that and also knowing that I couldn't do that easily at the University of Hamburg with that major, I actually put pen to paper and I wrote an application to spend the last year of my master's degree in Australia. So I applied for a grant from the German Academic Exchange Service to pay me or to fund like my last year of research towards my master's degree at the University of Adelaide, and they did. And so I flew over to study this little guy there, Chelidactylus nicropes, the magpie perch, which is a very abundant reef fish that is found around Australia. And I got to dive around this island that you see on the right-hand side of the slide, West Island, which has the field station of Adelaide University. And my thesis was really investigating the effect of prey number and density on predator foraging, predator being that fish. And it was a really important time for me. I had to improve my English quite a lot. And it was, I would say, definitely a period of quite immense personal and also academic growth. And we would spend weeks on this island, often underwater, collecting data. And I've chosen those two photos because I think they actually quite nicely show what was going on during that time. So on the left-hand side, you see me wearing my purple dry suit, holding a spear gun in my hand with a very nervous smile. That was because West Island was an island not just with cold waters, but also we had a big seal colony there and lots of sharks. And so me going in the water to spear this little fish that I was studying was not an easy feat. I told myself that having a purple dry suit would hopefully help to make sure that the sharks knew I was no seal. But also you can see that living on an island for a while, you develop a bit of a unique sense of fashion. And this is me here, probably still in my pajamas, but sorting out set-speared fish samples in formalin tubes. So that was really my time there in Australia. And it marked, I would say, the first really big, significant chapter of my scientific journey. And Sean Connell, he really taught me during that time the ropes of experimental ecology. And I got my first publication from that time. You see me here with a seal, in which we were able to show that it was really prey density that was most important for influencing predator foraging behavior. Well, then came a bit of a reflection point because, well, I should say that actually, I should say it a bit differently. Diving was in the cold waters around West Island and being surrounded by seals and sharks really set me up quite nicely for the next chapter of my scientific career, which was to join Kendall Clement's lab. And you can see he also had a good sense for fashion, and basically being a diver was almost, mandatory for joining his lab, particularly at that time. Now, the focus of my PhD project was to really look into the ecological factors that were associated with speciation in New Zealand triple fin fish. And this is a really amazing group of fishes in New Zealand. Many million years ago, one triple fin arrived in New Zealand and has then diversified into many species, around about 26 different species. And a lot of them are found all around New Zealand. They have the highest diversity here. They're also New Zealand's most abundant reef fish. So if you jump anywhere in the water and you open your eyes underwater, you probably see one. And we were really interested of what has actually caused this great diversity. A lot of them were overlapping. So what has actually caused that? Kendall at the time, he had a Marston grant to investigate that. and I was fortunate enough to be awarded a top achiever doctoral scholarship on top of that. So we were well funded, and then we were basically spending probably the next 3 years diving around New Zealand all the way from the Three King Islands down to Stewart Island. And during that time, I observed well over 20,000 fish underwater. Fabulous time. I learned a lot. And just in short to say, what we've really found by doing the research for the next years was that we are dealing here with an adaptive radiation that was triggered by an ecological opportunity, meaning that there was a lot of unoccupied space around New Zealand, and when the first triple fin came and was able to diversify, there were so many different habitat spots that it could move into. And so what we have in New Zealand is actually really remarkable. We have these different triple fin species that occupy quite different habitat types. We have some world records in this country because triple fins are found all around the world except Antarctica. But in New Zealand, we have the only estuarine triple fin and the only pelagic triple fin. Fabulous time. Kendall was an amazing supervisor, and I learned a lot. And we got 8 publications from my thesis, and thanks to Kendall, I also got the Maren Wellenreuther Prize for Best Ecological Thesis for the year. So after that, I had a bit of a moment where I really needed to sit down on a bench and think about my next step, because I really loved marine ecology, but I was also deeply aware that doing research in marine ecology is often quite pricey. You often need a skipper, you need to go diving, boat. And so I thought, why don't I become a terrestrial ecologist? Truly, you only need like a field car, petrol, and a butterfly net. And I know that's very simplified, but it appealed to me, and I did that. So I applied for a grant for a postdoctoral position to work on insects. And not any insects, damselflies, beautiful damselflies. And I moved all the way to Sweden, to the University of Lund, to study these Calopteryx damselflies. They're found over most parts of Europe near freshwater bodies. And what I was really interested in was to understand how climate is inducing range shifts in these insects and how that in turn is impacting species interactions. And so I was working on 2 damselflies at the time, Calopteryx virgo, this really dark species there, and Calopteryx splendens. And you can see, pointer's probably not working so well, but you can see that they have different ranges. So one is found further north than the other. And what I knew from being based in Lund, which is region 1, I knew that they were able to hybridize, but at a low rate. And so we were really interested is, given that Calopteryx splendens, the one with the wing spots, is currently expanding northwards, what does it mean in terms of hybridization rate when actually it comes into contact with Calopteryx virgo that haven't actually encountered females of this other species before? What does it do to the hybridization rate? And so what we did is that we sampled the species at 2 more points, and we looked for the likelihood of matings between them to describe the hybridization rate. And so we did that at the range margin, which is area 2. So that was absolutely the most northern point where Calopteryx splendens was found and was already quite rare. And then, that's why I cast there, we caught splendens in region 2, put them in a car and drove them up to the area they were expanding into. And we were looking at the hybridization likelihood there. And how did we do that? So how did we actually look at the likelihood of hybridization? So we caught females, and when we had the female, we would actually attach a cotton string, like make a little thread, we would make a little tie around the thorax and the abdomen of the female, and then we would attach that cotton string to a bamboo stick. And so you have this bamboo stick in your hand, and the female is flying at the end. And you can go to, like in the video where you saw the males are flying near the rivers. They have territories there. So you move your female with the cotton, with the bamboo stick over there, and you have the female fly right in front of the male. And you can see if the male wants to mate with her. So you're reporting that. So that's what we did for a long time. So we're catching females, tethering them to bamboo sticks, and then walking along the rivers, recording hybridization or mating likelihoods. And this is what we found. So we found, as I said, region 1 in Lund was low, at the range margin significantly increased, and then even further. So it really showed that these climate-induced range shifts can really have an impact on species interactions and species mixing as well. Now, I should also say it wasn't all easygoing. Being a postdoc in yet another, or being in yet another country wasn't easy, and being a postdoc was also not easy. At the same time, when all of this was happening, I also started a family. I had my first and second child. There was definitely ongoing pressure on us to publish. When I looked around at Lund University to further my career and to take on some extra training, a lot of it was only given in Swedish. And I definitely felt, or I had at least a moment, where I felt a sense of isolation. And I took this all in, but of course, the way I am, I do like to spring a little bit into action. And this is why I have this photo here, because, you know, I took this in, thought about it, and then I asked myself, well, what can you do about it? And so there are a few things that I did, and I want to briefly reflect on them. I put my hand up, and I became the organizer of the seminar series at Lund University, and that was great because it gave me access to budget to invite researchers from all around the world, and it really helped me to build my own network. I also joined the steering committee of the Lund Postdoc Society, and with this I was able to actually organize training for international students. I also decided that I wanted to become a docent, which is equivalent to the UK title of a reader. And I enrolled in weeks and weeks of pedagogy training, which was really helpful and got me to think a lot. And then I also joined the Female STEM Network, and shortly after became the coordinator of it. And last but not least, I put my hand up to proactively contribute to teaching. I will touch on the last 2 points a little bit more. So as I said, I joined the Female STEM Network, became the coordinator, and that was fabulous because with this I was able to really organize a lot of networking events, I organized conferences, and I got to meet a lot of people, but I also started writing about it. I wrote a book chapter, and I also organized a special issue that was entitled Women's Contribution to Basic and Applied Evolutionary Biology, which was quite a success. We had heaps of contributors. You can see them here on the front page. And I also got to write an editorial for it with Sally Otto that was entitled Highlighting the Changing Face of Evolutionary Biology. And I had contributions in that editorial from some of my scientific heroes like Rosemary Grant, Mary Jane West Eberhard, and Josephine Pemberton. And I also passed on my learnings, and I talked about it at several other places or other universities, such as Macquarie University in Sydney. But also, while I was doing a lot of the pedagogy training, I got thinking about course design and course concepts and all of that. And as I said, I was a bit frustrated that a lot of the things were only given in Swedish. But I also noticed that there were some real gaps in the curriculum at the time at Lund University. And so I actually designed a course. So I wrote a course concept, applied at the faculty for funding, and they funded it. And I want to and the course was all about science communication, because I noticed there was nothing that was being taught at Lund University around that particular topic, yet I thought it was probably quite important. So this was an extremely rewarding experience. We got funded for several years to do that, and it was a 1-week intense PhD course. It was open to students across all of Scandinavia, and every year we were fully booked out and we were teaching that, and we were really giving these students a communication toolkit, and we had journalists coming and actors and bloggers and all these great people. Very rewarding experience. Well, but of course my research life also continued at the same time. So I got another postdoc at the time, and this time a Marie Curie postdoc. And I should say that at this part of my career, I sort of started to add to my ecology and evolution background genetics, and that has stayed with me ever since. So the question that I was looking at for my Marie Curie postdoc was really, what maintains genetic polymorphisms, which is a really fundamental question in evolution. And I was still working on damselflies. And here I changed model systems a little bit. So here I'm now working on a Schnurra elegans, which is a species that is characterized by a female limited genetic color polymorphism. It's controlled by Mendelian locus in a single dominance hierarchy. And you can see the 3 female morphs here on the slide. You've got an androchrome and fuscens and a fuscens absoluta morph, and they differ in color. So now, what is maintaining this female color polymorphism? Because in any population that you're looking at in Europe, they are polymorphic. All 3 females are always there. And what we realized, so the team and I, what we found is that this polymorphism is maintained by negative frequency dependent selection because of male mating harassment. Just to explain that a little bit more to you, so what happens in these damselflies is a little bit akin to predator-prey dynamics, where predators will learn what is the most common prey in a year, and they will form a search image and then go disproportionately for that prey. Now in this damselfly, they will learn which color morph is the most abundant in a year, and they will disproportionately go for that morph. And because they have repeated matings and they're forced matings, it is extremely, it reduces the fitness of the morph. And so in the next year, that morph will be less common. So if a male would go for androchromes because they're the most common morph, in the next year they're probably less common, and then in the next year, other morphs are more common. And so you have these cycles over time that is really maintaining this color polymorphism in nature. And just to say a few more words about this male mating harassment, this is something very common in damselflies. As I said, males force copulations, and there are repeated copulations by males. And here you're looking at a gene expression plot, a volcano plot, because looks like this. You've got females on one side, males on the other. And we are now looking at genes that are differentially expressed between males and females. And if you look at it, you see the most important ones, pheromone detection. It's probably related to mate searching or something like that. But then you see in all of these blue dots, these are actually immunity genes. So that is because these matings are very stressful, and they often actually incur in injuries. And why is that? This is the penis of an Schnurra elegans male, and they have things like appendices such as brushes, hooks, grabbers, and spreaders. In damselflies, because they have repeated matings, when a male is mating with a female, the first thing he will do is try to remove sperm from previous matings, and then you need these sort of appendages. But it often leaves injuries. They also have sexually transmitted diseases, so females try to avoid males in this species. They ideally just want to mate once, hence the polymorphism and the cycles. I got for my work on damselfly species an award from the Swedish king, like you heard. It was quite a humbling experience to go up to Stockholm, to the palace, and to meet him, and to receive this award. And it was really towards contributions to biodiversity research. And it came really at a time when my particular research on genetic polymorphisms was sort of increasing and continuing. And I got also promoted to associate professor at the time. I started my own group, and I got a bigger grant from the Swedish Research Council to look at genetic polymorphisms in another model system, inversion polymorphisms actually this time. And I'm looking at this other model system right here. I guess you can't really see it because it's quite small, but I'm here on the Lofoten Island chain looking at rotting seaweed. And my new model system was actually this. It's pretty hard to see probably, but you see maybe some things flying. You have to go quite close. So these are seaweed flies. They live in rotting seaweed. They are a lot less charismatic than damselflies. But you know, I always like to get my hands on numbers and data, and so they suited me very well. And most importantly, they had this really interesting genetic polymorphism, an inversion polymorphism on chromosome 1, which included a lot of genes and had these massive phenotypic effects on males. So not females this time, right? With this Schnurra elegans, we had these color morphs. Here, we're actually having a big genetic polymorphism, and it's mostly impacting males. It impacts males on many traits, but the most important one was actually size. So you can have either the alpha or the beta inversion, and if you have 2 times the alpha, you're 9 millimeters in size, And if you have 2 times the beta, you are 3 millimeters in size. And heterokaryotypes, alpha betas, are 6 millimeters in size. So that's a 3-fold size difference. I mean, just imagine that in humans. It's massive. It's massive, and it has huge consequences. So we were, again, trying to understand what is maintaining this genetic polymorphism in nature. Because like with Ischnura, you could go to any population, it was polymorphic. So what is maintaining it? And I should say, even though we were studying it in seaweed flies, these inversion-encoded polymorphisms are actually really important across the tree of life. And here, just to show you some good examples where inversions encode really important traits or ecomorphs. For instance, inversions can encode different mating systems, as you can see in white-throated sparrow or ruffs, or they can cause phenotype-genotype associations, as seen here by Temema walking sticks or Numata longwing butterflies, or they can even cause these really behavioral ecotypes, like seen in fire ants or rainbow trout. But I was working on seaweed flies, but really a model to understand what is maintaining these big, genetic polymorphisms in nature. And we did do some work in Europe, and we were looking at these inversion frequencies across space. So you see a map of Europe there, or part of Europe, Scandinavia, and you see in these pie charts the different inversion frequencies. And you can see it's polymorphic in all populations. You also see that there's a slight decline of beta declining towards the Baltic Sea. Inclines are always classical signatures of natural selection in action. What we did during my Swedish Research Council grants was really to expand this research to North America, because the fly is also found in North America. And we sampled 8,000 kilometers of the coastline and looked at the inversion frequencies. And again, absolutely polymorphic, right? But no cline. We looked everywhere. We looked for salinity, latitude, temperature, anything, nothing. So we were really puzzled, like what is actually maintaining this genetic polymorphism if there's not such a strong link, like clinal link with environmental variables. And so we did something that you can do very well with insects. We did an experimental evolution experiment. Now I won't go too much into it because I don't have the time. But we basically did a 10-generation experimental evolution trial on this fly, where we were able to sort of disentangle the growth phase from the reproduction phase. And we were able to look at inversion effects of these different phases. And we were able to show that when it comes to the growing phase, that actually being yellow, being a beta, that they have much better survival and faster development in that situation. And that is really important when you're in a resource-poor environment, like wreck beds that are very thin, there's not a lot there, that rotting seaweed, or like a wreck bed that gets washed out to sea because of the tide. In those situations, being beta is best. However, it is much better to be gray when you are in a resource-rich environment, plenty of wreck bed, no rush to develop fast, because if you are alpha, you are much better at forcing copulations, here as well, and also the females are much more fecund. And so we are dealing with a situation where we are having a life history trade-off that is basically maintaining this polymorphism in nature, so balancing selection. After this came a bit of a reflection point again, because my research was going really well. We were really getting into it. We got good grants. I had students. But then my partner at the time, Damien Moran, he got a job offer at Plant and Food Research, a permanent position. And then after being interviewed, I also got a permanent position offered. And that's almost like a dream come true, right, for scientists, having like suddenly 2 permanent positions. But it was outside the university, and I thought this was really scary because I'd never been outside the university. And so at that time, Richard Newcomb and Rochelle Constantine were in Lund. They were on their sabbatical. And I still vividly remember walking with Richard in the park and literally saying to him, "Am I going to commit academic suicide?" And he said no. And I'm really glad he did. Because honestly, I do think now, which is like more the last chapter of my life, it was, probably the most rewarding part of my scientific career, because what this move allowed me to do was really to connect my fundamental research with, all the way to an impact, to an applied side. And that was something that took a little while to learn, still learning it, I think, but it's been extremely rewarding to learn that and to be able to actually translate science all the way to something that matters to industry, to people. And so at first, of course, you can imagine moving countries and changing is not easy. So at first I said I want to be 50-50. So I stayed at Lund University for 50% of my time. I still had students, research grants, I finished that, and then went for 50% to PFR in Nelson. It was a hell of a commute. I did that for 3 years, and then I fully based myself in New Zealand. And shortly after, an opportunity came up to join the joint graduate school between Plant and Food and the University of Auckland, which I then joined in 2018 as an associate professor, and I've been here ever since, and it's been fabulous, because with this I've had really access to great minds, good colleagues. I was able to teach and also meet students, so it's been a really interesting part. And going to Plant and Food in Nelson, I was able to continue my sort of more fundamental interest in research, and I was given a lot of freedom to do so. And just one slide on that. So I really, my sort of fundamental research on genetic polymorphisms has really stayed with me, particularly focused, as I said, I sort of moved into inversion polymorphisms, but then a big focus on structural genomic variants and how they really matter in ecoevolutionary dynamics. I've worked a lot on that, but over the years I've also sort of moved more and more to New Zealand relevant examples for my research, of course. One example here is the SNAPper genome, version 2 that we've just published, and that shows you sort of on the outer ring the 24 chromosomes, and the inner ring show you the different types of structural genomic variants that we were able to explain, to describe, such as deletions, insertion, duplications, inversions, and translocations all the way in. And we have been studying how they impact ecoevolutionary dynamics and the phenotype. So that has been going on, but I will not focus on those aspects for the rest of my talk. I actually want to move on a little bit more to the applied side now, because as I said, that has been extremely rewarding. And so I want to talk a little bit more of how I have been using my knowledge in ecological genomics to really trying to achieve a little bit more of an applied outcome and really achieving some impact. And so one thing I want to talk about is fisheries management, and the other one is aquaculture. So first, how can we actually use genomics in New Zealand to inform fisheries management? I give you 2 examples. One is really to review the stock structure of species that are otherwise difficult to study, such as hoki, deep water. Or how can we use genomics to inform seafood traceability? And here I will use snapper as an example. So a little bit, this is actually a project that came to us by industry walking through the door and saying, we would like you to look into this, we do not know. At the moment, hoki is our most important commercial deep water species. It's currently managed as 2 stocks, but industry was wondering, is it actually, are there actually 2 stocks? Are we managing it properly? Or are there maybe 3? Are there more? Is there 1? So what we did is that we worked together with industry. We sampled 12 sites around New Zealand and also 2 in Tasmania, and got 510 individuals, and we got whole genome sequences for all of them. And then we plotted it, and this is just a but it shows you 2 clusters. And this is the whole genome information from all of those 510 individuals. And you clearly see all of the hoki from Australia cluster and all of the hoki from New Zealand cluster, which indicates, we did additional analysis to that, but it indicates that New Zealand hoki are genetically one, or they mix quite a lot at least. And so we have been since communicating that with the New Zealand and Australian fishing industry to inform their management. Another species here, snapper. This is an example of how genomics can be used to inform seafood traceability. Now, hoki, they migrate quite a lot. They go all the way to the subantarctic islands and all of that, but snapper moves a lot less. And so when you have a lot less movement, you can get all this genetic structure. And so we really wanted to understand a bit more about snapper structure. And so we had 10 sampling sites and again 350 genomes. And this is what you get for snapper. Quite a different situation. You actually get a southern and a northern cluster. And you see there's quite a bit of sort of flow between the two. They're not like completely separate clusters, but there are clusters nevertheless. And if you put this pattern on a map, it looks like this. So you've got, as I said, the northern red cluster and the blue southern cluster. And you have these mixing zones around Mahia Peninsula and Cape Reinga. But what is actually really cool, if you look a little bit deeper, you also see that from the different sampling sites that we have, that a lot of them contain local genetic substructure. So all the ones that are encircled with blue actually have local genetic, genetic signal. And so what you can do with that genetic signal, you can use that for seafood traceability, because if you have a fisherman that got quota to catch something on the west coast and is landing that, you can actually check if it's truly from the west coast or maybe instead from Gisborne. So that's extremely powerful. And for that sort of work, I got the Hamilton Award in 2018, which was great because it really opened doors to a lot more and having your science being heard and being recognized. And I got to speak at Parliament shortly after, talking about things such as, the use for genomics and how we can really use it for sustainable management. We don't use genomics at all for fisheries management in New Zealand. And I think there's a real opportunity to do so. It also opened the doors to contribute to some sort of more decision-making and working groups. There were several, but I give just one example. For instance, I was part of the working group that was initiated by the Prime Minister's Chief Science Advisor, Juliette Gerard at the time, called The Future of Commercial Fishing in Aotearoa, New Zealand. And so we put this report together, and this was really, really a rewarding experience to be able to contribute to that. And this is kind of what we put sort of these sort of ideas in the report, what could a future look like? This is what we're doing on the left-hand side at the moment. This is how we're doing fisheries management. We're using all the length and abundance data and things like that from trawls. But actually, if we add to that genetic data, we can really get improved insights into stock structure, how many individuals are there, connectivity, to really model the genes to ecosystem response of a fishery. So this is a vision, but I think it's a good one. So the last part that I want to very briefly touch on is also how genomics can be used to inform aquaculture, which is really important for New Zealand. We're an island nation. We have a lot of opportunity. And so we've been focusing on a number of species, but the species I want to highlight is the Australasian snapper. It's a species we've been selecting because of such huge importance in New Zealand. It's a really important customary species, but also one of our big inshore fishery species. It's also important for recreational fishing. And it also thrives in the warm waters around the North Island, which is important because at the moment our only finfish species, Chinook salmon, can be found around the South Island. So we've been really lucky because a lot of this work that I'm showing you, whether it's fisheries, genomics, or aquaculture, we've been doing that together with industry, with Maori, and that gives us often access to resources that make the research so relevant. For instance, here for the aquaculture research, we are using a sea pen that we are leasing from Naitao Seafood, and they are sponsoring a lot of the work as well. And we can just scale things up and run them at a semi-commercial level, which has been quite important. And just to show you one of my last graphs, this is just to show you how we have been using genomics to accelerate breeding gains in the Australasian snapper. You see in blue our selected strain and in green an unselected strain, just to give you a bit of an idea of what that can look like and what you can achieve with fast and improved selection. And it doesn't just increase the growth rate, in our case by 30–40%, but we are also getting, when we are comparing the improved and the non-improved snapper line, we're getting much better survival and improved FCR, so food conversion ratios. Now, my last slide, really. Just some quick reflections, because it's not easy to put all of this together from the last, I don't know, 20 years or so. But looking back, a few things that I've learned, some reflections. Well, often people have said, oh, why do you have so much energy? How do you do this? And I truly think that if you do something that really awakens your curiosity and gives you meaning, that you do have a lot of energy. So I would always say to everyone, stick to your gut. Don't do something because other people tell you to do that or because it's trendy. Honestly, like, follow a little bit your own path. Definitely also noticed, like, it's okay to be ambitious. It's not really about us, what we're doing here. It's okay to have these big ambitious projects and have a long view, and definitely put on your piece of the puzzle, help others with theirs. I also, I'm a strong believer of that we all need to sometimes leave the ivory tower. I mean, I find it sometimes really uncomfortable and would like to run the other way, but I sort of push myself and say, come on, let's do this. And if we all do that, that's really important, because I think making our research matter is so important, and we just sometimes have to do it, however we want to, but a little bit. I also really think it is quite important to always be open to a challenge and to be able to critically discuss this. Maybe that's my father coming through, but I think we all have our own ideas and our own ideology and our own beliefs, but you have to be able to move that to the side and look at it and really critically and openly discuss this. It's very, very important. And yeah, last but not least, have fun at work and definitely celebrate success. Being a scientist is not always easy. It looks great when you put it together like this and when I hear other people talk about it, but there have been lots of steps that weren't so smooth or that were hard or where things didn't go that way. And so when they do go well, definitely celebrate them. Really, really important. And that's my last slide. I talked a lot about myself now, but we all work in teams, and everything I presented today is big teamwork. You know, I mean, kudos to everyone involved. I had heaps of people supporting me over the years, and not just my scientific colleagues. Also, of course, family and friends that have always been there supporting me. Thanks for my Nelson whānau being here, and also Auckland people. So thank you so much for being here and supporting me. Thank you.